between two heterozygous, a male and female. So genotypes came out to be AA, big A, little a, big A, little a, little a, little a. So 1/2 came out to be to have the Angelman syndrome, and 1/2 did not. Half unaffected. And that was just, the unaffected was because of the genomic imprinting. Part B was a cross between a heterozygous male, I believe. It's big A, little a, little a, little a, big A, little a, little a, little a. So because the genomic imprinting came out to be 100% Angelman. For part C, we got 0 bar bodies because the XIST would be inactive and the T6 wouldn't count it because it was inactive. And for part D, we got 2 bar bodies. For part 2A, let me read these real quick. We got the sex determining region Y is an example of a holandric gene. For part B, we got the MYC2 gene, which produces a cell surface protein, and it's an example of a gene found on both the X and the Y chromosomes. For part C, we got half normal and half dwarf. For part D, it was in the sex influence pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other sex. For E, we got genomic imprinting and dosage compensation. For F, we got T6 and XIST. And for part G, we got when reciprocal crosses give identical results, the alleles that control the trait in question are located on an autosome. And when reciprocal crosses give different results, the alleles that control the trait in question are located on the sex chromosome.
All right, so for question 3, is that our favorite system?
Yeah.
All right, so for question 3, we're going to scroll down real quick and take a look at this. So we're told that a woman's father has the OTD, which is an X-linked recessive disorder, and then the mother is homozygous dominant. So we know that the genotypes for part A are going to be, we're going to denote a lowercase a as the X-linked recessive, and then the mother is going to be homozygous dominant, as was told in the problem. For part B, it says if the woman has children with a man who does not have OTD, what is the chance that their first child will have a son with OTD? So we know that based on this cross, if it's a woman, the only possible genotype that she could have if she's unaffected, the only genotype that she could have is be heterozygous. And then if we know that the husband is not affected, then he's going to have dominant. So all we're going to do is just do a simple test cross. So if we do a test cross, we're going to see that the chance for a male to be affected is only 1/4 because it's an X-linked recessive gene. So this right here is only going to be a 25% chance. Part C, it asks, identify the genotype of a male with whom the woman could produce a daughter with OTD. So the daughter is going to have to be homozygous recessive if she's going to have that disorder. So if we know that the mom is heterozygous, the man, the father, husband, or whatever you want to say, has to be affected because there's going to have to be X's that are both going to be recessive to give the daughter that type of genotype. And then part D, consider the cross. So they're going to ask us to cross the mother and the father. So the mother is heterozygous, the father is affected, and then it says what is the genotype and phenotype of the offspring. So we're going to do a simple test cross. You can see where this is going. So as you can see, 1/4 is going to be a female carrier, 1/4 is going to be an affected female, 1/4 is going to be a unaffected male, and 1/4 is going to be an affected male. Just going to scroll down.
So for number 4, we're supposed to find out where the beard genes are. So for part A, we are supposed to suppose that a cross produces 3 beard, 1 no beard phenotype ratio among the male offspring, and then so we're supposed to figure out what the genotypes and phenotypes of the parents are. So from this, from the part above, we can link that a uppercase B is a dominant allele for males, and the lowercase b is a recessive allele for males. And we can associate a uppercase B to be recessive in females and a lowercase b to allele to be dominant in females. So the first cross to get 75% of males with beards and 25% with no beards, we're going to cross parents that are homozygous. And you're going to get big B, big B, big B, little b, big B, little b, little b, little b. And from that, you get 75% with beards and 25% with no beards for males. And then for part B, we're doing the exact same thing, except for females, we're finding the ratio of 1 beard to 3 no beards. So we're going to also cross a heterozygous male and a heterozygous female, or mother. And from this, you're going to get big B, big B, big B, little b, big B, little b, little b, little b. And from this, you also have 75% no beard, and then 25% will have beard because of the homozygous recessive gene. For part C, I'm going to erase these real quick. For part C, we're supposed to find one that produces 1 beard and 1 no beard with females. So we're going to cross a father that has big B, little b, and then a homozygous recessive female. And you're going to go big B, little b, little b, little b, big B, little b, little b, little b. And from that, you're going to have 50% of females that have no beards, and you're going to have 50% that have beards with females. For the next one, you're supposed to do 1 beard, a ratio of 1 to 1 no beard and beard for males. So you're going to cross a homozygous recessive male and a heterozygous female. And from that, you're going to get little b, big B, little b, big B, little b, little b, little b, little b. And you have 50% of them that have a beard and 50% to have no beard.
All right, so for 5, let me erase this real quick. So yeah, I can't talk today. Part 5, there we go. So involves X-linked inheritance or reciprocal crosses. So in cats, orange coat color is determined by the little b allele. Black coat is determined by b allele. Heterozygous. So we'll write that down. So we know big B is equal to black. Little b is orange. And then a heterozygous is going to be tortoiseshell or torch. All right, so question A says, suppose you cross an orange male cat to a tortoiseshell female cat. What are the genotypes and phenotypes of the progeny? So if we know that the male is orange, we're going to put little b crossed with a mother that is a tortoiseshell, correct? Yeah. All right. So the mother's going to be like this. She's going to be heterozygous. So all we're going to do is just do a cross. All right, so we see that 1/4 of the females are going to be tortoiseshell, 1/4 of the females are going to be orange, 1/4 are going to be males with a black coat, and 1/4 are going to be males with an orange coat. By the cross, part B asks, male tortoiseshell cats are very rare. What is the genotype? So they're going to have some type of disease where they're going to have 2 X's and also a Y. So they're going to have 2 X's that are heterozygous along with a Y, and that's why they're so rare, because that doesn't happen in nature, or happens very rarely. Part C, it asks about the fruit fly, about scalloped wings and normal wings. So it's going to ask what happens when reciprocal crosses are done. So what we're going to start with is we're going to start with a male that is affected, crossed with a female that is a true breed for the homozygous dominant. If we do crosses, what we'll get is we'll get 50%. So we'll get all the females will be normal and the males will be normal. A reciprocal cross is when you switch these. So a reciprocal cross, we're going to have a male that is unaffected crossed with a female that is affected. So if we do the cross, we're going to have 50% of the females are going to be unaffected, but this time 50% are going to be males that are affected. So this means that it is a X-linked recessive or a sex-linked chromosome because the reciprocal crosses are different. In part D, it asks assume that the scallop phenotype is an autosomal recessive. So what we're going to do is we're going to just cross and say, let's say the male is homozygous dominant and the female is homozygous recessive or affected. The only option you can get is this heterozygous. If we do a reciprocal cross where we just switch these, you're still going to get the same, where it's going to be a heterozygous. And when both of the results are the same, that means that it's an autosomal trait, and therefore you can determine what kind of trait it is.